The adapter hypothesis and tRNA. The adapter hypothesis states that nucleic acid wouldn't by itself be able to differentiate between the amino acids to attach to nucleic acid, so an adapter molecule would need to be used with the assistance of a protein that could distinguish between amino acids. So therefore, Crick, who came up with the adapter hypothesis, actually predicted the idea of tRNAs, which are the so-called adapters. How does tRNA work as this adapter then? Well, T in tRNA stands for transfer. So transfer RNA, so it transfers the amino acid that its three-letter codon is specifically linked to. It does this by first attaching itself to its amino acid, which is done by first activating the amino acid with ATP, creating aminoacyl-AMP, and then to attach the AMP leaves and the tRNA attaches to the 3' end. Then the tRNA travels to the RNA, in which it will eventually match up its three-letter codon with the codon in mRNA. mRNA is being read from 5' to 3'. It starts with the Shine-Dalgarno sequence, which is important for ribosome binding. A ribosome is made up of two subunits, large and small subunit. mRNA will attach first to the small subunit with its Shine-Dalgarno, and then eventually tRNA will become charged with FMET, and in coherence with IF2, GTB will bind with the initiation codon in mRNA. Once that is complete, all the initiation factors leave and the large subunit is allowed to connect. There are 3 sites on the ribosome that involves reading the mRNA and tRNA. There's the A site or activation site where the tRNA first connects with the mRNA, the P site where new peptide bonds are formed, and then the E site or exit site. This will then continue until a whole protein is formed and the gene has been read. An alteration that could affect the adaptability of tRNA is that it isn't charged and no amino acid is able to attach. Then when the codon sequence on mRNA was read, by this tRNA, no amino acid would be added to the chain and the potential of building the wrong protein would occur, which would be bad. The consequence is that the cell would be disformed or die without the appropriate protein to help it function.